I'm Andrew Helmstetter from the French Foundation for Biodiversity Research, and today I'm going to talk to you about the state of state-dependent speciation and extinction models, patterns in angiosperm macroevolution. One of our goals as evolutionary biologists is to uncover the origins of diversity. We know that species diversity is unevenly distributed across the tree of life. And much research goes into uncovering the causes behind this uneven distribution. In particular, we often want to know how species character traits, such as floral nectar spurs, viviparity in fishes, or the relationship between plants and their pollinators can influence species diversification. Today I'm going to address these questions and themes in the context of angiosperms, a large group also known as the flowering plants, that contains more than 300,000 species and encompasses 140 to 240 million years of evolutionary history. Humans have a close relationship to angiosperms. For example, their importance in agriculture, so understanding their evolution is paramount. The principal question we wanted to ask was, which traits have shaped angiosperm diversity? To answer this question, we turned to a set of methods that could help us understand trait-driven diversification, the state-dependent speciation and extinction models, or SSE models. These models are based on a time-independent birth-death process, where diversification rates depend on a state of an evolving character. The original model was the binary state speciation and extinction, or BISSE model, which concerns two character states, each with their own speciation and extinction rate, as well as rates of transition between these states. The SSE family of models are some of the most popular models used today to investigate species diversification, and BISSE has been extended to incorporate multiple states, to allow for quantitative traits, to focus on geography, and to include hidden states. We synthesize the results of studies using these approaches by collating all studies that use SSE models on angiosperm clades, about 135 different papers when we first collated them in 2020. We aim to figure out which papers identified state-dependent diversification, which traits they investigated, and the characteristics of the datasets they used to make their inferences. To aid in collating the data, we developed a small R package called papier-mâché to help us extract information from papers. Papier-mâché can automatically assign papers to categories based on the frequency of term use. For example, if a paper mentions dioece 5 times, it's probably about dioece. It also allows the user to interactively pull out chunks of text based on combinations of keywords, highlighting the relevant information as shown here. The goal of this package is to make gathering information for synthesis studies more efficient, and if you're interested, it can be found here. So, what kind of traits are associated with changes in diversification in angiosperms? This bar plot shows different trait categories on the x-axis and the number of times SSE models were used on traits in these categories on the y-axis. The proportion of the bar that is green is the proportion of models where trait-dependent diversification was inferred. For example, traits relating to geographic range were commonly tested, but associations were not often found. Conversely, for traits relating to flower morphology, life form, or fruit morphology, trait-dependent diversification was detected more often than not. We can zoom out, classifying traits into larger categories, which reveals that we are often interested in traits relating to reproduction, where there is a 50-50 split, and that vegetative traits, those related to plant growth, morphology, and physiology, are often a source of trait-dependent diversification. Angiosperms are very diverse, but what groups are we, as researchers, most interested in? This tree map shows the different orders studied, scaled by the number of papers where the focal clade belonged to each order. As might be expected, we're often interested in studying species-rich orders like Asparagales, which contains the orchids, or those that are particularly relevant to humans, such as the poales, or grasses. To develop upon this theme, we plotted the number of species in an order on the x-axis versus the number of studies on that order on the y-axis. It's revealed quite a linear relationship, where more effort is being put into larger orders, as we might expect. But this also points out that there are groups that are relatively understudied, such as the Asterales, where there are lots of species, but not so many papers investigating their trait-based diversification. The data we collated also allowed us to explore trends in how SSE models themselves are used. For example, how has SSE model use changed over time? This stacked bar plot shows publication year on the x-axis and the frequency of model use on the y-axis. Different colors on the bars represent the different types of SSE models used in each study, and the black line is the total number of studies on angiosperms per year. Since our inception, there was a rapid increase in SSE model use, largely driven by the BISI model, until a conspicuous slump around 2015, where after approaches including hidden states such as HISI rose in popularity. This slump corresponds to the publication of several key papers criticizing SSE approaches, such as this one by Roboski and Goldberg. And this is perhaps a good example of how such critical papers can shape how we do research. We then wanted to see if there was a relationship between the characteristics of datasets used with the SSE approaches and whether we were detecting trait-dependent diversification. So, here are two density plots with the number of tips on the x-axis. The color density plots represent SSE models for which trait-dependent diversification was inferred, and the gray density is where no effect was found. So, as might be expected, SSE models used with larger trees more often inferred trait-dependent diversification. A similar pattern is found for the age of the phylogenetic tree used. An absence of trait dependence is more often found when we're using younger trees. Tip bias, here defined as the ratio between the frequencies of the most and the least common character states in the tree, appear to have similar densities in models with and without an effect. We also examined trends in the number of markers used to create the trees that were used with SSE approaches, a proxy for the quality of the tree. We found that there was some evidence that using larger numbers of markers more often yielded trait-dependent diversification. Lastly, and perhaps most interestingly, we found that there was a marked relationship between sampling fraction and trait-dependent diversification, where models in which a smaller proportion of the focal group was sampled were inferring trait dependence more often. This is some cause for concern, and we're currently delving deeper into the data to try to unravel the causes behind this trend. Given that we found several interesting patterns between the input data and the outcomes, we decided to go wild and see if we could predict whether we would infer trait-dependent diversification using only the input data, without actually running an SSE model. We used an approach called extreme gradient boosting. This is a machine learning approach that uses a set of weak prediction models, usually decision trees, to perform classification. We're by no means experts in these types of approaches, but thought it would be an interesting experiment. We trained the model on 75% of the data and tested it on 25%, and found that we could predict whether we would infer state-dependent diversification at between 70 to 80% accuracy. Variables in the model are plotted here based on their relative importance. Those continuous variables relating to the tree, its sampling, and tip states were the most important, but there were also some categorical variables, such as whether you're using FISI, working in asparagales, or testing traits relating to habitat that help the model predict the ultimate result. In conclusion, we found that intrinsic traits related to reproduction, growth, and morphology were often involved in trait-dependent diversification. That there exist knowledge gaps in angiosperm groups such as asterales, as well as understudied trait categories that could be useful avenues for future research. We also found that the input data was both important and informative. Particularly sampling fraction needs to be considered carefully when conducting SSE approaches. I was tasked with going through and extracting the results from all of these papers, and I can tell you of the 135 or so studies in the dataset, there were about 135 different ways of reporting SSE results. So we plan on detailing a set of best practices to do so. Linked to this, to help verify the data we've collected, if you've used SSE models and angiosperms, we might contact you to ask you to check what we've taken from your study, so please keep an eye out. With that, I'd like to thank the FRB CESAB for funding, my colleagues in the diverse group for their work on this project, particularly my supervisors Sylvain and Jos, and thank you for listening.